Scientists have discovered the first new antibiotic in decades, giving hope of stopping the relentless spread of drug-resistant diseases. Teixobactin was found in the soil around Boston in North America. It's unique because it works differently to other bacteria, meaning it could be the key to combating some of the world's deadliest infectious diseases.
Professor Kim Lewis has achieved what other scientists have tried to do for decades but failed. He and his team in Boston have discovered a new antibiotic which can kill serious infections in mice without any apparent resistance.
It's our best compound so far of the 25 that we discovered. Teixobactin is different from other antibiotics in that we do not see any resistance development in pathogens, and that is unique.
Teixobactin works by targeting parts of the cell that don't mutate, unlike other antibiotics which attack the cell's protein. The discovery has raised hopes of fighting drug-resistant infections, including tuberculosis. In 2013, 1.5 million people died from the disease, almost all of them in the developing world.
We're casting around desperately for new drugs that are less likely for resistance to develop. So to have a new class is very important for TB.
More importantly, for the developed world, Teixobactin could help combat the deadly superbug MRSA, so prevalent in hospitals.
It's now the case that the antibiotics that we've used are so common in the community that we see these resistant staphs occurring in people who've not been in hospital as well.
Teixobactin's discovery relied on a tiny device called an iChip, which allowed scientists for the first time to grow uncultured bacteria in their natural environment instead of the lab. The Boston team believes the same process could be used to develop dozens more antibiotics. But it could be years before the research ends up on pharmacy shelves. Anne Barker, ABC News.